Before I start, could I just have a quick show of hands? Who's here for the first time, your first time at OTS, or you've been working in the oligonucleotide field for less than 2 years? That's great. Okay, so this lecture is for you. I'm delighted to be able to address this topic today, and especially at this conference where I think we see really an amazing display of some of the excitement that we all share around the prospect for using oligonucleotides as therapeutics. And I just have a snapshot from this review that came from David Corey earlier this year. It's really impressive the variety of applications that oligonucleotides potentially have. And here, of course, I'm really focusing on intracellular opportunities, differentiated from the extracellular opportunities that we heard about in Paloma's talk. So you add it all up, and oligonucleotides really point to a future of genomic medicine. And it seems that oligonucleotides should be the ideal tool for accomplishing that, with one significant exception, and that is they are extremely unattractive as drug candidates. So the liabilities are well known: poor metabolic stability, rapid elimination. Paloma mentioned the sequestration binding to various proteins, competitive uptake by non-target cells, and of course, cell membrane repulsion, the fact that these compounds are really not well suited to entering cells, and yet that's where the majority of the targets are that we're interested in engaging. So the question comes up, how can delivery technology help? Well, one way of viewing this is to consider the concept of an RNA delivery vehicle. Most of my illustrations will be with RNA today. And in a simplified view, here's a simple framework. You start with a cargo, identify the goal, might be an intracellular goal. Draw from previous experience. Mark mentioned that in his opening lecture, the value of starting with something that you know, and then maybe incorporate other features or ingredients to get it to work. Add the cargo, use as directed, and after you get to where you want to go, you exit the vehicle, and there you are. There's nothing inherently wrong with viewing it this way. The problem, though, is that this view tends to kind of take on a Hollywood-like perspective. So, for example, what might the cargo be? Well, it might be Einstein, the dog. And what might the goal be? We're looking at really ambitious goals, intracellular delivery. What about an ambitious goal? Delivering Einstein 1 minute into the future. For previous experience, DeLorean, impressive technology, especially in the mid-'80s. Maybe we can start with that. Add a flux capacitor. Put the cargo in the vehicle, strap in Einstein, follow the directions, 88 miles per hour, and boom, back to the future, 1 minute, you've accomplished your goal. So this is the problem with RNA delivery vehicle as a concept, is that people tend to have a very Hollywood-like view of it. And what I'd like to share with you today and give you examples of to convince you that addressing delivery hurdles is a much bigger problem than just looking at delivery technology, which oftentimes people use when they're really talking about formulation technology. And that's not enough to overcome these hurdles. I advocate a more holistic view, looking at the full spectrum of drug R&D tools to solve this problem, especially tools from basic chemistry and biology, really the full arsenal of drug development tools, that overcoming these delivery hurdles is like regular drug development, only harder. It's not easier, it's harder. So what I'd like to do this morning is step you through examples from each of these areas, and of course there's some overlap. We'll start with a formulation technology example, and then I'll share a chemistry example and some nascent examples that are very much, from my perspective, biologically influenced. And we'll end on the biology note. So our goal, ambitious goal, the illustrations that I'm sharing are drawn from sRNA, and initially we'll focus on delivery to liver, which I think is arguably the easiest in this challenging arena to potentially achieve. And so to get to that target, of course, the drug candidate needs to survive the blood, it needs to distribute to the target organ. Within the target organ, it needs to reach the intended cells, typically hepatocyte. It needs to be taken up into the cell, typically by some endocytic mechanism. Then, if a vehicle is used, it needs to be unpackaged from that vehicle and escape the endosomal vesicle. And then the cargo needs to reach RISC, the RNA-induced silencing complex. So that's a lot of hurdles to overcome. And one important question is, well, how will we know that we achieved our goal? How will we know that we actually delivered to where we intended to deliver? Well, I like to see quantitative measures to illustrate that. So, I think the most direct would be an mRNA knockdown by quantitative PCR. So that's a direct measure of message knockdown. But even more direct than that would be the quantitation of guide strand in AGO2 by an immunoprecipitation. And that technique is something that we worked on at Merck and is published. By the way, I'm going to tweet all the references, so you don't need to write them down madly. You can just look at my website or my Twitter feed, which is in the attendee list. I'll do that later today. I put on here PNA hybridization. That actually just came out, Advanced Online NAR, a couple weeks ago from Arrowhead, an alternative approach, but the same end in mind, direct quantitation of guide, loaded, and RISC. That's a very direct measure of did we achieve our goal. What I would consider to be less convincing would be, for example, race data, which is an amplification assay, so you can amplify a needle in a haystack. It doesn't, it's not totally useless, but it's not really telling you how well did we do our job. Even lower on the list, from my perspective, would be tumor response, which is a very indirect measure, or any other pharmacodynamic response, where that response could be due to a non-specific effect, and you can really trick yourself that thinking that you actually delivered your cargo, and when in reality you did not. Just some examples. Here's a nice data set with antisense, so not sRNA, antisense from Ionis, a paper they published about 7 years ago, showing a very nice correlation. If you look at the axes, I think you can see that. The liver concentration of the mipomersen, this is in mouse, with plotted against the liver ApoB mRNA levels. And so you can see that the more drug is in the liver, the less mRNA is detected. So that's an illustration of what I'm talking about here. Examples. I'm going to share an example of a formulation technology. I'm going to share an example of a application of chemistry technology. Then we're going to take a little bit of a detour and talk about extrahepatic, because these first two areas we're really just focusing on liver delivery. So I think it's important to consider extrahepatic delivery, which can be systemic or local. So systemic would be a systemic administration, IV sub-Q, where you're targeting something other than liver. Local is where you're directly administering to the target tissue itself. Then we'll cover the biology examples that I promised. So formulation technologies, here just a few. The one in the upper left, I believe that was a publication from Judy Lieberman quite some number of years ago, and then transferrin cyclodextrin came from Mark Davis. We're going to be talking about the one on the right, which is a lipid nanoparticle platform, and there are various manifestations of this. The SNALP, which is the top figure there, is from the Tecmara technology, now known as Arbutus. I've shown some EMs of lipid nanoparticles from Merck. Dicerna, Silence, Novosome, Pronai, all of these companies have platforms that are similar to what I'm going to briefly describe to you. So the lipid nanoparticle technology involves the packaging of siRNA duplexes in what are termed fusogenic pegylated cationic lipid particles. And the particles are nanoparticles, typically around 100 nanometers in size, depending on the conditions. And they're made by a simple T-mixing. The particles form spontaneously due to entropic effects, and are formed by mixing an siRNA solution with an ethanolic lipid solution with lipid components that are shown in the figure there. And so the question is, well, how did they work? Here's some data showing that they work very well. By this measure that I proposed as sort of the report card measure, a quantitation of guide strand in AGO2 from an immunoprecipitation pull-down assay from livers from mice. So the bottom panels there are from the reference shown from luciferase siRNA in lipid nanoparticles, similar to what I just shared with you. And there are actually two versions of this. The one on the lower right there is the liver tissue level, so that's sort of comparable to the mipomersen data up top that you saw a moment ago. And then on the left is the actual quantitation, not just of liver levels, but actual AGO2 associated luciferase siRNA, and a very nice correlation with the knockdown based on bioluminescence. So it works. How well does it work? Well, if you look really closely, you can see that the liver levels in picogram are picograms per mg, so it's like 10,000-fold less in the liver compared to mipomersen, which on the surface, that sounds great. That's a lot more potent. But you can also quantitate, kind of the report card. If I put, 100 siRNA duplexes in, how many got to risk? Better not look at it that way because it's less than one. Here's sort of a visual assessment of how efficient is it with the Psi-5 label, and you can see that 30 minutes, 2 hours, liver glows up. And after 24 hours, there's basically just a whiff left behind. So this, of course, is a pretty nonlinear response, but it gives you a clear picture visually that not a lot is left, and yet the knockdown response is very strong after 24 hours. With the immunoprecipitation, you can actually calculate how much got to risk. And depending on the exact formulation and conditions, it's a very small amount, like 0.01%. Certainly less than 0.05%. Very, very small fraction of administered siRNA that actually makes it to the liver with this technology. So the technology works as intended, and it's a very potent drug, so it gets the job done, but it's very, very inefficient. So I do recommend the paper shown at the top there, which from my former Merck colleagues, quantitation of what I call where do the eggs fall out of the basket. And it's actually very interesting because in that paper they showed not just losses due to endosomal escape inefficiency, but also due to inefficiency in risk uptake following endosomal escape, and that's something that hasn't received a lot of attention. The paper on the bottom right goes into details on endosomal recycling, vesicular recycling, as sort of an unproductive path following uptake of these particles. So a number of things happening, contributing, that remain to be elucidated. So how can this be improved? Well, I think, with my vision of viewing this as a drug development project, really improvement often comes about through hard systemic mechanism-based structure-function analysis and progress based on mechanistic insights. What are the mechanisms? How did this really work? I said it was fusogenic lipids, but that's just words. So here we've got some cartoons to go with the words. The simple fact is that the theory behind these systems is more than 15 years old and hasn't really been updated. It's an area that, in my judgment, is very much unexplored. These mechanisms actually come from plasmid work, so it's not directly even the same systems. Two works that I'm citing here are work from Cyrus Safina from UC Santa Barbara, where he focused on lamellar structures and identified the importance of surface charge density in governing the actual escape of the contents following uptake. And then the other work that I'm citing there on the right is from Frank Shoka at UCSF, and Frank's focus was on hexagonal phase formation and the propensity of certain lipids to form hexagonal phases that have a greater tendency to fuse, and that this would presumably be responsible for enhanced endosomal escape. The challenges with these are that when you're looking at a 0.01% roughly of the payload that's actually getting the job done, that's the fraction where you care about the mechanism. All the other stuff, well, those are unproductive mechanisms, and it'd be nice to know something about them if you could kind of shut them off. But the mechanism that really counts is not really taking place in 99.99% of your samples, so it's not easy to study this. So we've heard about inefficiency challenges. We've heard about the incomplete understanding of mechanism. We don't have time to go into some other challenges: species variability, nanotoxicity, manufacturing challenges. There's a lot to it, but I'm going to move on, and we're going to talk about a chemistry illustration. I'm going to cover this fairly quickly because it will be the subject of some of the, whoa. Wow, well, I can see the screen. Yeah, this is fine. Okay, that's looking promising. So in the hopes that that's going to warm up, I can see the slide here. I'm going to move forward. So chemistry examples. The chemistry example that I think is the most interesting for the purposes of today's lecture is something that's been presented at this conference several times before, and you'll see later in the week. That is the GalNAc conjugates that have been advanced farthest along by Alnylam. And the slide that you can't see is actually a slide from Alnylam's website. Mano has shared this at this conference in the past. And that involves targeting the acetylglycoprotein receptor using a triantennary structure of the sugar GalNAc. That receptor is extremely attractive for targeting hepatocytes because of its high expression level, its high uptake rate, and its rapid recycling time. So it's a very unique situation of factors that make that an extremely attractive target, mechanism of targeting. And so in this case, I've included it as a chemistry example because it's a conjugate of a chemically modified sRNA duplex with the triantennary structure of the sugar attached to it. So obviously a lot fewer components and not really a formulation technology. This is formulated in, I believe, a simple aqueous buffer. So that, of course, is work from Alnylam, and I encourage you to go to Vasant's lecture. But I also wanted to include this slide, which I also took from Dicerna. This is from their website, a slide that Bob Brown, who's also presented at this conference many times, shared at Asia Tides back in February. And it's just to point out that the concept of GalNAc targeting of the acetylglycoprotein receptor actually goes back many decades, and there are a lot of very interesting work that has been done over those decades in order to get this to work. And research in this area continues. Continued advances, a paper that came out in 2015, where the actual display of the GalNAc is being further optimized in some cases for benefits of ease of synthesis, greater binding potency, whatever. Just basic I would call it medchem optimization of the basic approach. And of course, Ionis has also applied this concept to antisense, where they published in this paper a couple of years ago a tenfold potency improvement. So a lot of very interesting studies coming out of what I would consider to be a more chemistry-based approach. So let's shift gears and talk a little bit about extrahepatic delivery. So those examples we just showed, of course, were focused on hepatocyte. And the first on the list, which is very much in the news because of the approval just last week of Sarepta's eteplirsen, is their lead, eteplirsen, which is based on PMO chemistry for a neutral backbone to dial out some of the inherent features in RNA that make it hard to deliver. And eteplirsen, for those who don't know, is actually administered IV, but of course the target in Duchenne muscular dystrophy is muscle. And some of this actually gets to muscle, and there is a certain level of dystrophin that is expressed in the truncated form by the the intended exon skipping mechanism. Very inefficient, I think, by any measure, and of course this has been a fairly controversial topic in both the public media and in the technical press, and probably at this conference. Other examples for extrahepatic, I think the most significant category would be local delivery, and local delivery is often viewed as sort of the low-hanging fruit, that if delivery is just so hard to achieve, these hurdles are so hard to overcome, then why not just put the drug where it needs to go and not worry about this, and at least get a product out there and worry about the hard stuff later. And this has been a strategy by probably most RNA companies, and Paloma mentioned the anti-VEGF, aptamer, so not an intracellular target, but aptamer that became the product Macugen, a fairly successful product. One of the few other RNA products approved, Vitravine from Ionis, was also administered intravitreally. Cerna, later acquired by Merck, had a partnership with Allergan with a VEGF, I believe it was a VEGF receptor-targeted siRNA. Quark is in the clinic with siRNA in the eye. Alnylam RSV was administered with a lung target. I believe it was administered nasally. More recently, Marina with direct instillation in the bladder in one of their programs. Gut examples in gut with Ionis, again, in their Elicoforsin, ICAM targeted antisense. More recently, Nogra partnered with Celgene with their SMED7 targeted Mongersen antisense program. And then CNS, which I personally view as the most exciting, and I'm certain that Stan Crook will be discussing this in his keynote, which I'm sure you will all find quite stimulating. So a lot of activity in local delivery. The problem with local delivery is that oftentimes the low-hanging fruit is really, turns out not to be that rewarding. And in fact, many of these products have not been very successful, and I would argue, don't have time to go into the details, but more than half of the things on this list, I would argue that the preclinical data supporting them was all based on nonspecific effects. So the problem with local delivery is if you're kind of in the mood to cut corners and get things done as quickly as possible, it's easy to cut corners on the mechanistic part, and that can lead to disappointment in clinical translation. If you take a step back, how does the field look? These data are a couple years old, but the point is, and this is from, I don't see the reference on the bottom there, this is from the RNA.dk, the Danish website. And if you look at all the RNA stuff in the clinic, the vast majority is simple aqueous formulations. So I think that tells you something about the state of the field. Now, I'm going to close here, and am I allowed to go over like 3 or 4 minutes since the power went out, Mark? I'll take that as a yes. I'm going to close and speak to you a little bit about that third bubble, the biology bubble. And the simple fact is that many of the unanswered questions, what I would say the most intriguing unanswered questions are very much biological in nature, and we could spend all day adding to this list, but just a few that I tossed on here. ASGPR, GalNAc has been very fruitful. What else is out there? Is there anything out there that will function at this level? That's a real good question, and I haven't heard a good answer to it. And no is not a good answer. That's the answer we don't want to hear. How can endosome escape efficiency be increased without increasing toxicity? I didn't draw your attention to it, but in the GalNAc example, there is no dialed-in mechanism of endosomal escape. The stuff just kind of leaks out, and enough of it leaks out to get the job done. Why is that? Well, typically mechanisms for increasing endosomal escape, they cause membrane damage and lead to toxicity. So therapeutic index is often a very serious problem. I'm highlighting biology mechanisms and trafficking, and yet the tools for studying those are largely in vitro cell culture tools, studying cell trafficking in tools, not all of them, but most of them. So our growing understanding of trafficking and what impacts it and how formulations and designs can leverage it, how relevant will they be to the in vivo situation? Well, I guess we'll find out. So a lot of unanswered questions. If you look at, here we're still at the cartoon level, but, just from a more biology point of view, this review from Kai Simmons about 6 years ago, and, there are a lot of different pathways into the cell, and the composition of the vesicle following uptake depends on the mechanism by which you got there. And so any mechanistic attempt to overcome the barriers that result, it's kind of shooting in the dark if you don't really even know how you got there. And the simple fact is that these mechanisms are probably all happening simultaneously at some level, and they vary from cell type to cell type and from species to species. So it really is a much more complex picture, and I think that picture has yet to fully impact research in the delivery arena. So a couple of examples, and then we're going to close, and I may even end up closing on time here. I'm putting these in the category of what I would call biology influence, and technically they're more pharmacology influence, but just to show you what I'm getting at in terms of how having a biology point of view can change your thinking. The first example on the upper left is from Arrowhead, and it's actually, what I would call a deconstructed, more of a formulation chemistry approach with their dynamic polyconjugate that came from Mirus. And with Arrowhead is currently in the clinic with ARC-520. It's also been presented at this meeting. And now they're delivering two components, actually three components, because there are two RNA components. But the active component is the two RNAs, siRNAs. And then there's sort of the pharmacological tool component, what I would call it, is this melatonin-based delivery agent. And so they deliver that. It's got the sugars to target at hepatocytes, and that kind of makes the hepatocytes more, leaky. And then the RNA gets into the hepatocytes. So you're really leveraging the pharmacological impact of that first agent. So you're causing biology to happen, and thereby getting your cargo in. Similar concept, these two papers that I'm showing here, one from Marino Zariel at Max Planck in Dresden, the other from Rudy Giuliano, looking at enhancers of delivery from chemical library screens, just screening small molecule chemical libraries, looking for compounds that would enhance RNA uptake, again, by some pharmacological mechanism. With the screening approach, the mechanism isn't really known. That would maybe be figured out post hoc after identifying hits. So these are just some of the approaches that are more of a biology point of view. Then with my last slide, I want to share another frontier in this arena of more biology-influenced delivery, and that is exosomes. So if you're not familiar with exosomes, that's been a very hot area, especially the last 10 years. And this paper on the upper left came out in 2007. It was not the first paper, but certainly a widely noted paper that drew a lot of attention to exosome-mediated transfer of RNA in vivo, that this is, at some level, a natural mechanism for RNA transfer in living organisms. And that, of course, raises the prospect, can this be leveraged to commandeer to deliver RNA cargo for therapeutic purposes. And a couple, I just cherry-picked a couple papers here, but there are others out there. Matthew Wood's paper, work also that's been presented here in the past, on delivery of siRNA to mouse brain by the injection of targeted exosomes. And then a paper that Anastasia, and I can never pronounce her last name, so I always just say Anastasia. That just came out. I think it's in Advance Online, or maybe it just appeared in print about 4 weeks ago, using exosome-mediated delivery of siRNA for Huntington mRNA silencing. So a couple of really interesting examples. And then this last example that I'll close with came from Novartis with some academic collaborators, where they looked at exosomes as an actual mechanism of entry into cells. And I love this title, exosome surf. How can you not read a paper that starts that way? So just to highlight the frontier, the edge of knowledge that we don't really understand RNA trafficking in vivo very well, and that there's an emerging understanding that will have a lot of implications for delivery, and I think ultimately point to some very exciting applications. So with that, I'd like to close.